Hey, good afternoon, everyone. Thanks for joining the webinar today. So as introduced by GM, my name is Caroline, and I'm the Senior Application Manager for Standard BioTools in charge of the SomaScan platform in this region. So just a brief overview of Standard BioTools. At Standard BioTools now we do offer a diverse portfolio of products spanning the proteomic as well as in the genomic space shown in this slide here. So we're going to talk about the SomaScan assay today, but we also provide the flow cytometry, which is our CyTOF XT system. And in terms of the spatial proteomics platform, we do have our Hyperion XTI as well as our genomic workstation, which is the biomarker X9 system offered by Standard BioTools. So a brief introduction to the SomaLogic technology, right? So SomaLogic, which developed the SomaScan assay, was founded in year 2000. So actually the technology has been around for 20 years, and then we have merged with Standard BioTools about 1.5 years ago, and we are all now under Standard BioTools as one company. Then over the years, with the development of the SomaScan assay, we started with our 1000 protein plex panel, and we try to improve the assay by increasing the number of targets that we can detect over the years with our development of 4K assay, 5K, 7K, and the current latest panel is the 11K assay. So the core to the SomaLogic technology relies on this reagent known as SOMAmer reagent. SOMAmer stands for slow off rate modified aptamers, right? So as you could see on the image on your right, the green and the purple part is the SOMAmer reagent. And what we make with these SOMAmer reagents is that in that purple region, right, we make chemical modifications to this single-stranded DNA that mimic amino acid side chains that are known to be involved in protein-protein interaction. So through these chemical modifications, right, it really enhances the binding of the DNA to the proteins. And through this specific shape recognition of this SOMAmer reagent with its specific protein partners, right, they have a very high binding affinity. So when we started with selecting of the specific SOMAmer reagents that bind to its protein partners, right, we start with a 10 to the power of 15 libraries of DNA sequences with this modification. We then do selection one purified protein at a time, go through multiple rounds of selections with different stringent conditions, and then we will be able to select SOMAmer reagents that shown to bind to its specific protein targets. So here is an illustration demonstrating that the way the aptamers and binds to its proteins are similar to how antibodies engage proteins, right? So there are publications that shows that the shape complementary index between aptamers and antibodies are very similar, as you could see from the slide here. So the way that aptamers and antibodies bind to the epitope on the proteins are in a very similar manner. So they are both affinity-based platform. And aptamers are a lot smaller in size compared to antibodies. They are only about 8 to 15 kilodalton compared to antibodies, which is 150 kilodaltons. So this slide shows a high-level overview of how the SomaScan assay works. All right, so in image A is the SOMAmer reagent shown in purple. Right, each of these SOMAmer reagent has 3 different moieties attached to it. It has a biotin molecule to enable the reagent to bind to the structured avidin beads. It has a photocleavable linker which enable us to cleave this protein DNA complex of the beads, and then the fluorescence tag, which enable readout on the adjuvant microarray system. So in image B, upon binding of the samples with the SOMAmer reagent, you see that there are specific protein binding that happens, and because we selected for this slow off rate DNA aptamers and with high binding affinity to its protein target, it sticks together with the protein targets for a very long time. Then in cases of non-specific binding that happens between the proteins and the SOMAmer reagent, because they are non-specific proteins binding, they dissociate within seconds to a minute. We then cleave these DNA and protein complex of the beads using UV light, which is placed in the liquid handler that we use to run the assay. This whole workflow is almost fully automated, right? The only manual step is to placing the samples and consumables onto the workstation. Then during this cleavage step, right, in image E, we also add another level of specificity to our assay, which is by adding the poly-N ionic competitor shown in green here. So this poly-N ionic competitor, again, are single-stranded DNA, but they are not selected for any specific protein. So they have a very low binding affinity to their proteins. The aim is to act as a competitor to the SomaMer reagent to bind to those non-specific proteins that has been released into the solution to prevent these non-specific proteins from rebinding to the SomaMer reagent, and they get washed off during the workflow. So at the end of the assay, what we are interested is the fluorescence tag SomaMer reagent, as shown in image G. We will release these fluorescence SomaMer reagents from the protein complex using a high sort elution buffer. We then collect this fluorescence tag SomaMer reagent and hybridize it onto the adjuvant microarray readout platform, which consists of the complementary sequence to the SomaMer reagent. So the SomaScan assay is a relative quantification assay, right? Basically, what you get at the end of the data is the relative fluorescence unit generated for each target and in each of the samples that you have run, and then you can compare the fold change differences between the groups of samples that you are interested to compare to. So one of the key challenge with a proteomic assay is the ability to detect wide dynamic range of protein concentration in matrices such as human plasma and serum. As we know that in human plasma and serum, right, there are highly abundant proteins such as albumin to the very low abundant targets such as cytokines. So with the SomaScan assay, how do we achieve these 10 logs of dynamic range of different protein concentration within human plasma and serum? So we have run thousands of human plasma and serum materials. We do understand what is the endogenous protein levels like in this type of matrices. So we have actually worked out the best dilution protocols that we should use for human plasma and serum. So as you could see here in the image on your right, so for human plasma and serum protocol, we do dilute these samples into 3 different dilution bins, shown here, 1 in 5 dilution, 1 in 200, and 1 in 20,000 dilution. And then at the bottom of the table indicates the percentage of SomaMer reagents in each of these different dilution bins. So 80% of our SomaMer reagents are targeting the low abundant proteins in the femtomolar to picomolar range, and then 18% of our SomaMer reagent are targeting the mid-range expressing proteins in the nanomolar range, and 2% of these SomaMer reagents are targeting the highly abundant targets in the micromolar range. And through this optimized dilution protocol, we are able to capture these 10 logs of dynamic range of protein concentration within human plasma and serum. Okay. Regarding the specificity of our SomaMer reagent, we do understand that DNA aptamers is something new to our customers. So during our R&D development, we did spend a lot of effort verifying that the SomaMer reagent is indeed pulling down the specific protein targets that it's supposed to be. So the process for selecting of the specific SomaMer reagents to a specific protein target is called selects. So basically, we have this 10 to the power of 15 DNA sequences with modifications, and then we select 1 purified proteins at a time. So these SomaMer reagents with a specific shape, will then find its binding partners to the specific proteins. And then at the end of this select process, right, the SomaMer reagents that show to binds to this protein will then be eluted and amplified. And then we will do downstream validation, such as looking at the sensitivity of these SomaMer reagents, understand the binding affinity of the SomaMer reagent. We will also screen against highly abundant proteins to make sure that the SomaMer reagent only binds to the specific targets and do not pull down those abundant proteins as well. So we have done a pull down assay to check the size of the proteins, and then we also did further confirmations of these proteins from the pull down assay using a mass spec platform. So because we have run thousands of plasma and serum materials, so we do have the performance characteristic of each of these SomaMer reagents as shown here in this second image, right? So we have the performance such as the precision of each of the SomaMer reagent, what is the CV like for this SomaMer reagent in the human plasma and serum, what is the signal to noise ratio, limit of detection, F statistic, etc. And then for human plasma and serum, we have also built a population reference for the normalization purposes. So over the last 20 years, there are already over 1,000 publications using the SomaScan platform. So there are a lot of publications demonstrating that customers have used other methods to confirm that this Soma reagent is indeed pulling down the targets that they are interested on. And these include, for example, using ELISA technique for confirmation, mass spec technique, or cis-QTTL association. So here is an illustration demonstrating that we also further tested our specificity by looking at 1,580 SomaMer reagents with known homologous proteins. So we tested 4,200 homologous proteins against these 1,580 SomaMer reagents to look at whether these SomaMer reagents, what is their binding to their closely related protein partners. So as you could see that 85% of these tested SomaMer reagents do not show any cross-binding to its homologous protein. 6.8% of the SomaMer reagent do show cross-binding, but with 10 times weaker affinity compared to its protein targets. And then 7.5% show similar cross-binding to its homologous protein. So all this information is available on our online menu tools at menu.somalogic.com, which means that if you search for a particular target and then you look at the characterization column of these particular targets, we will indicate whether this SomaMer reagent has been tested for homologous protein testing and whether it has shown binding to its protein partners. So here is a summary showing the number of orthogonal validations, number of SomaMer reagents that have gone through 1, 2, or 3 additional orthogonal validations. So all our SomaMer reagents have gone through primary validation in-house in our R&D department, and then these are just the number of reagents that have orthogonal confirmations through publications. So in terms of content-wise, our latest panel is the SomaScan 11K assay panel. As the name implies, it allows you to detect near 11,000 proteins. And then we also have the SomaScan 7K assay, which allows you to detect about 7,000 proteins in the single aliquot of 55 microliters of human plasma or serum. So here is a summary of the total number of measurements between both the 7K and the 11K assay. So total number of measurements indicates the number of SomaMer reagents that we have in each of these panel. And then unique human protein measurements means that the unique proteins are based on UniPro ID that can be detected in both of these panel. So with 7K assay, you can detect 6,713 unique proteins. And then with the 11K assay, you can detect 10,070 unique proteins based on UniPro ID. And in terms of performance-wise, right, so you could see that both our 7K and the latest 11K panels have pretty consistent performance of a medium CV of around 5% in human plasma and serum material. And then the signal-to-noise ratio, percentage of content with a signal-to-noise ratio of above 2 is at 99%. And then the sensitivity in buffer, which is the LOD in buffer, is in the femtomolar range. So a comparison between both the current SomaScan assay with our nearest competitor, which is an antibody assay, right? So shown here are the percentage of targets that we are able to detect in each of these different major biological pathway. You could see that with our SomaScan panel, we detect 30 to almost 50% more targets in each of these known biological pathway compared to the competitors. And in terms of looking at some specific disease areas such as cardiovascular disease, inflammation, oncology, and neurodegenerative disease, we have increased 30 to 40% content-wise in this important disease area in our current 11K assay. So one of the key advantage with the SomaScan assay is our reproducibility. Our assay has a very low medium CV of 5% in human plasma and serum, and then we are actually leading the industry standard in terms of the reproducibility. And what does this mean? So what does this medium CV of 5% means to you? Right. So shown here is an illustration that we created bioinformatically to understand the number of additional samples required to to remove or account for any technical variation with the assay. So if you run an assay with a medium CV of 5%, like the SomaScan assay, right, you only need 8 additional samples to remove these technical variations in your study. But if you run on a platform with a 20% CV, right, the number of additional samples required to account for these technical variation will increase from 8 to 126. So it means that, you know, the more reliable or reproducible the assay is, the less samples that you need to be tested. And here is a comparison with our SomaScan assay performance over the years compared to the Olink assay, right? So we started with a 1K panel, and then we developed multiple versions of assay over the last 20 years, from 1K to 5K to 7K and to the latest 11K panel. As you could see, that our performance remains fairly constant of around medium CV of 5%, even though we add significantly more targets to our current panel. So with the Olink antibody assay, right, so they started with a 96 targeted panel where their CV are pretty good at around 10%, right? So the y-axis is 1 minus CV percentage. So here is a CV of around 10% with their 96 targeted panel. When they increase the target from 96 to their 3K panel, right, their CV increased to around 15 to 20%. And then with their latest Olink 5K panel, their CV actually increased to 30 plus near 40%. So what this it shows means that with the antibody affinity platform, they clearly have a challenge in increasing the number of targets that they have in their panel without not being able, they have to sacrifice the performance of their assay, you know, by increasing the number of targets in their current assay. So another key advantage of the SomaScan assay is the ability to correlate your data together, right, without any bridging sample. And this is particularly true for human plasma and serum because, as I mentioned, we have a reference population that we use for normalization. So we will compare your data against this reference population, and through this normalization strategy, we have shown that, you know, customers are able to combine their data running at different time points together without having any bridging sample. So here are just some illustrations showing examples where samples run with different cohorts. For example, in our dementia prediction risk test, we use studies from the ARID Visit 5 cohort and the ARID Visit 3 cohort, as well as the VLSA cohort. We are able to combine this data together where there are actually no bridging sample across these different cohorts. And then one of the organ aging publication in 2024 that is published also demonstrated that they are able to combine data, the SomaScan data from 5 different cohorts together, as you've seen here at the bottom of the slide here. So this publication uses 2 cohorts run on the 5K panel and 3 cohorts on the 7K panel, and they are still able to even combine the data with different versions of assay together. So in terms of the types of samples that can be run on the SomaScan assay, although the SomaScan assay has been heavily validated using human plasma and serum, but over the past 20 years in our lab in Boulder, right, we have actually run multiple different sample types on top of human plasma and serum. Human urine and CSF are commonly run samples in our lab. So the assay also work for mouse and non-human primate plasma and serum materials, cell lysate, tissue homogenates, and cell culture condition medium, etc. And the requested volume for each of these different sample types is indicated in this table here. If you are using human plasma and serum, all you need is actually just 1 aliquot of 55 microliters. So we do indicate 2 aliquots of 55 microliters here in the table, or 1 aliquot of 130 microliters to be sent to us. And the reason is the extra volume are used as a backup samples in case if anything went wrong, we can immediately repeat your samples without having you to resend your samples again. And so currently all these sample types are enabled on the SomaScan 7K assay as well as the 11K assay, except for human urine and human SLE samples where they are only enabled on the SomaScan 7K assay. Okay, so one of the most useful way to explore our menu to look at the targets that we have in our 7K and 11K assay is through these website menu.somalogic.com, right? So you just need to do a simple registration with your name and email. You should be able to access to these menu tools, and then you can search for the targets that you are interested. For example, here you can look at what is the amino acid range that we use for the initial select process to select for the SomaMer reagent. What is the performance of each of these SomaMer reagents using human serum and human plasma? So for example, the signal to noise ratio, what is the intra and interplate CV for these particular targets? And then are there any published orthogonal confirmation of these particular SomaMer reagents? All this information are available on our online menu tools that you can simply find the information that you want for the targets that you are interested in. So in terms of the data output, right? So our data output is in a text file known as an ADAT file. Basically, this is a text file that you can open up in Excel. So what you see here is that you have columns of each of the targets in our assay, and the rows are the number of samples that you have run. So basically, you will get the pre and the normalized RFU unit generated from the run. You can then compare the fold change differences between the groups of samples that you wanted to compare to. So with each of these ADAT file, we will also provide a quality statement report known as the SQS report. This is a report that tells you the number of samples that you have run and whether the assay passes our QC metrics for the run. So in terms of data analysis, we do have a web-based analysis tools known as the data diagnostic tools, where again you can do a simple registration on our website, and then you can actually import your ADAT files into this data diagnostic analysis tools, and then you can create volcano plots, box plot, do simple statistical tests, and then they will also pull down the list of the top hits using this software as well. And then if you are familiar with R or Python languages, we do have available R or Python languages or open source software for data analysis. So the key differentiator of the SomaScan assay is really firstly, in terms of the number of targets that we are able to detect, right? So our current 11K menu covered over 50% of the known human proteomes. So, and then the other key advantage would be the ability to correlate your data for plasma and serum material without the need of bridging samples. So you can run your samples now, and then if you have more samples to add on in the future, you are able to actually just add on the samples and combine the normalized data together without having need of bridging samples. Then obviously our assay are highly stable and reproducible with a medium CV of 5% in human plasma and serum. Our assay have a very wide dynamic range of 10 logs of dynamic range for detection of proteins in the femtomolar to the micromolar range. And in terms of the sample volume, if you're working with human plasma and serum, all you need is just 55 microliters of plasma and serum. And you do not need to do any purifications of proteins from the plasma and serum. Just spin down the blood, get the plasma, or let it clot and get the serum, and then send us 55 microliters of the plasma and serum material. Okay, so in the second part of my talk, I would like to go through some case studies to illustrate how you can make use of the SomaScan platform in different application areas. So I wanted to first show some platform comparison data that has been published when the last year as well as this year. So the first head-to-head comparison data was done by this ERIC study group headed by Joe Koresh. Right, this was published in 2024, where they actually run the same samples on both the latest SomaScan 11K assay and the latest Olink Explore 5K assay. So what you could see in this figure here in the orange bar are the SomaScan 11K assay, right? Y-axis is the frequency, which is the number of targets, and then x-axis is the CV. So you could see that most of the targets on the SomaScan assay has a CV of less than 10%, And in the blue bar is the Olink Explore HT 5K assay, where over 2,000 of their antibodies actually has a CV of 50% or higher. Then they are able to manage to save their CV if they fix their LOD values at a fixed value of LOD divided by 2. They are then able to save their medium CV of 36% down to 14% with this calculation. Then looking at just within assay correlation itself, right, on the SomaScan platform, our medium correlation within batch 1 and batch 2 sample is at 0.9, while for the Olink HT assay, the medium correlation is at 0.66. So how about the correlation between the SomaScan data versus the antibody data? What is the correlation like? So very often we do see this bimodal distribution in terms of the data correlation between the aptamers technology as well as the antibody technology. We do see targets that are highly correlated, and then there are also targets that do not correlate as well. And then based on the study that the ARID group has done, they try to find out what is the reason that, why these targets are not correlating very well. So you can see this the plot on the right, the reason that why that these targets are not correlating very well is due to the fact that many of these targets are actually below the LOD detection of the Olink Explore HT platform. So this year, February, there's another largest platform comparison publications that has been published by a company called Alkahurst, right? So they look at not just the affinity-based proteomic platform, the SomaScan and the Olink assay, but they also included 3 different mass spec platform and do a platform comparison between the commonly used proteomic platform in the industry. So shown here are the types of assay that they have used for these particular publications. In terms of the SomaScan assay, they use both the SomaScan 7K and 11K assay. And then for the Olink assay, they use the 3K and their 5K panel. For the mass spec platform, they have chosen the SEER Proteograph mass spec nanoparticle platform, where they use nanoparticle for protein enrichment. And then they have used the Biognosis True Discovery, where they deplete the highly abundant protein targets to enrich for the low abundant targets, as well as the mass spec gold standard, which is the Dermo SureQuant, which have internal standards to allow for absolute quantification for the comparison. So the first comparison is looking at the technical precision across the different proteomic platform, right? Shown here, as you could see that in terms of the technical replicates, right, they either run 2 or 3 technical replicates to look at the technical precision. SomaScan assay has a very tight bar here. As you could see, our assay are very highly reproducible, and then between the 2 different versions of our assay, the SomaScan 7K and assay as well as the 11K assay, we do see very consistent performance in terms of the technical precision. And they also confirm what the ARID group has observed with a very low medium CV of around 5% in their study. Then in terms of the signal to noise ratio compared to the other proteomic platform, as you could see here, the biological variability versus the technical variability demonstrate that the SomaScan assay has the best signal to noise ratio compared to the other proteomic platform. So looking at the percentage of proteins that are detected above LOD across these 78 commonly run plasma sample across the different platforms. Again, the SomaScan assay indicated here at the end of the graph show that we are able to detect the highest number of targets above LOD. And they also wanted to understand the biological relevant markers detected across these different proteomic platforms. They also include these clinical metadata such as age, sex, race, hematocrit level, total protein level, smoking status, as well as BMI, to look at the significant markers identified across these different platforms associated with these different factors. So as you could see, again, the SomaScan assay demonstrated that we are able to detect the highest number of significant markers compared to the other proteomic platform. So they also wanted to look at, in terms of the protein classes detected across the different proteomic platform defined by this Panther classification system, right? Here is a heat map illustrating the different protein classes identified across the different proteomic platform. Again, the SomaScan 11K assay demonstrated that we are able to detect the highest number of protein classes defined by the Panther classification system compared to the other proteomic platform. And in terms of the number of FDA-approved protein biomarkers across the different proteomic platform, SomaScan assay again demonstrated that we are able to detect the highest number of FDA-approved protein biomarkers compared to the other proteomic platform. So the take-home message of this particular study demonstrated that the SomaScan assay offer the most extensive proteomic coverage, right, in terms of the number of proteins or biomarkers that you can identify with our latest 11K assay. And also we provide the highest coverage of FDA-approved biomarkers compared to the other proteomic platform. Again, this group demonstrated this low technical CV of the SomaScan assay with the highest data completeness. And then they also mentioned about the importance of a platform selection will dependent on your research questions, right? So if you are interested in the broad biomarker discovery, there's no reason why you shouldn't use the SomaScan 11K assay. But if you are interested in certain applications such as looking at isoforms or post-translational modifications, then maybe mass spec is a better platform compared to the other platform. So the need for a multi-platform approach, right, is required, right, for our customers or researchers to gain better understanding into biomarker discovery as well as validation. So the SomaScan assay has also been used in multiple world largest population cohort studies as indicated here. So for example, China Kadoorie Biobank has run 4,000 samples on the SomaScan 7K assay, and then they are also looking into running more samples on the latest 11K panel. We have the EPIC study where they have run 30,000 samples with the SomaScan 7K assay, and the MESA study, which is the multi-ethnic study group that has run 15,000 samples on the SomaScan assay. And the bottom of the slides are other examples of the biobank or cohort studies that have made use of the SomaScan platform. So all these large biobank studies have generated millions of protein measurements to enable researchers to gain deeper insights into the disease that they are interested to look at. And I wanted to show some preliminary data based on the EPIC study where they have run over 30,000 samples on the SomaScan 7K platform. So EPIC stands for European Prospective Investigation into Cancer and Nutrition. Their main focus is to look into cancer. And these are samples that are collected across 10 different Western European countries at 23 different centers, and over the 20 years of this collection or follow-up of their longitudinal studies, right, there are individuals that stay healthy and there are individuals that develop cancer during the way. And the aim of their study is to try to identify biomarkers that can help for early cancer prediction. And the main reason that they have chosen the SomaScan platform is because they wanted a broad-based biomarker discovery. They want to cast a wide net, so they wanted to choose a platform with the highest coverage. So here are some preliminary data based on their data, right? So looking at breast cancer, right, as you can see on the volcano plots here, they are able to identify some of the known breast cancer biomarkers, but on top of those known biomarkers, they have also identified thousands of biomarkers that is currently unknown. And then they did a comparison between the different version of the SomaScan assay to look at, the number of biomarkers that can be detected, right, with the different version of the SomaScan assay as well as compared to the Olink assay. So let's see comparing the SomaScan 7K versus the Olink 5K assay, right? So if you they demonstrate that if you run on the Olink 5K assay, there will be a 59% loss of biomarkers discovery if you run on the lower plex panel. So they thought that, okay, maybe this is just specific to breast cancer. How about other cancer types? Do we see the same phenomenon as well? So they looked also into lung, stomach, and prostate cancer. And then so this time they compared to the UK Biobank data sets which run on the previous Olink 1.5K panel, right? So here showing that a comparison between the EPIC Biobank SomaScan 7K data versus the UK Biobank 1.5K Olink panel, right? The reduction in terms of biomarker that you can discover when you choose on the lower plex panel. So the next example is also a very recent publication done by Novo Nordisk, which is published this year. The aim is to look at the proteomic data to see whether this proteomic assay can provide insight into the mechanism of action of their drug, specifically semaglutide, that has been used for obesity as well as type 2 diabetes. So they have used this in their phase 3 trials, the step 1 and step 2 trials as indicated here on the SomaScan 7K assay. So these are individuals that are overweight and obese with and without type 2 diabetes. So here are the volcano plot demonstrating that they are able to identify for the 495 and 277 protein targets that are significantly affected upon semaglutide treatment. And then in both of these trials, and many of these proteins are known to be related to obesity as well as type 2 diabetes pathophysiology. Then they also look at the correlations across the 2 trials, and they see that there are about 79% overlap of biomarkers identified across the 2 independent trials. And what they also mentioned in their publication is that about 60% of these measured significant markers or biomarkers that they identified are only measurable on the SomaScan assay, but not on the our competitor platform. So upon adjusting for weight loss as well as the level of HbA1c, they are able to identify 47 and 15 proteins that are significantly affected by semaglutide treatment. Many of these proteins are implicated in a diverse biological effect, and then they are associated with a higher CVD risk. So they also make use of our cardiovascular Soma signal test, right? So this is an in-house proteomic test that we have built ourselves, which means that we have used the data sets generated on the SomaScan assay, look at markers that can predict the risk of developing cardiovascular disease in the next 4 years, and we call them the Soma signal test. So this is particularly on the cardiovascular Soma signal test, where they run this test on their data sets as well, and see whether the semaglutide has a cardiovascular benefit. So what you could see here in image B is that upon semaglutide treatment, there is a reduction in terms of the cardiovascular risk, which confirm what they have observed with their SELECT trial. So during their SELECT trial, they have also seen a cardiovascular benefits of this semaglutide treatment. Then they also then compare the data that is generated with the DECODE study, and the reason for using the DECODE study is because this is also done on the SomaScan assay, and then it also provides a comprehensive proteomic data sets for both the healthy as well as diseased individuals. So they look at the proteins that has been shown to be downregulated or upregulated on the DECODE data, and then how that with their semaglutide treatment. So shown here, for example, like in the neuropathic pain, where these proteins were shown to be downregulated in the DECODE data, but were actually reversed. Instead of being downregulated upon semaglutide treatment, they were actually upregulated. Right. Then combining the G1 studies from the DECODE studies assessing the genotypic associations for higher genetic risk of BMI as well as type 2 diabetes, right? What they see is that upon semaglutide treatment, it consistently reversed these protein expression patterns that are associated with a higher risk of BMI as well as type 2 diabetes. So the take-home message of this particular study demonstrated that the SomaScan assay are able to reveal some specific effect of semaglutide on proteins and different biological pathway. This gives them a take insight into the drug function, as well as produce additional insights into whether the drugs have additional other functions on top of just obesity related. Then they again demonstrated that the SomaScan assay are highly precise, which confirmed what others have found out, and also they make use of the cardiovascular Soma signal test to actually confirm the observations of these cardiovascular benefits of semaglutide that they have observed in the Novo Select study. So the next example is about the feasibility of actually developing a LDT test on the SomaScan platform. So OncoHost is a company where they actually develop a test known as the Prophet platform. This is their in-house test that they have run the validated samples on the SomaScan assay, and then use machine learning to develop protein signatures together with the level of PD-1 and PD-L1, and then to help clinicians to make a better informed decision whether the patients will be beneficial of just doing chemotherapy itself or using chemotherapy together with immunotherapy. So they have actually published this data. So if you're interested to understand their perfect platform for treatment of non-small cell lung cancer, you can take a look at their publication. But what I wanted to point out in this particular publication is that they again demonstrated the experimental precision as well as the experimental accuracy of the SomaScan assay. So in terms of the experimental precision, right, they two technicians run the same samples, and then they look at the intraplate and interplate CV. You could see that the intraplate CV is again at around 5% range, and then the interplate CV is also at the around 5% range that we have demonstrated. They then send the samples that they have run in-house to our Boulder lab to run, and then to look at the set-to-set comparison, and demonstrated 88% accuracy for all proteins. They also tested their computational precision, which means that how accurate is in their model in calling these samples to be perfect positive or negative, right? So they have run multiple samples with multiple replicates, and then run the models, and then you can see that out of the 14 samples, 13 samples predicted 100% accuracy. So overall, about 93% accuracy in terms of their computational precision. So this study shows that it is possible to develop a lab-developed test, right, using the proteomic data generated on the SomaScan assay, and in this case is a model that they can help to predict or better to help clinicians to make better decisions whether the treatment, what is the best treatment plans for the non-small cell lung cancer patients. So the next example is about a publications on the dementia signal test that has been published again recently, right, in 2025. So the dementia signal test is a 25 protein model developed in-house at SomaLogic, where we make use of the ARIC visit 3, visit 5 study to look at the models. So the model development is based on the ARIC visit 3 data sets, and then the validation is done on the ARIC visit 5 data set, as well as the BLSA data set, and then we also independently validate it on a Japanese populations by working with our collaborator in Japan. So here is a Kaplan-Meier curve showing the dementia signal test, where the tests are able to categorize the patient into the low risk, medium low risk, medium high risk, and the high risk. So you can see this wide dynamic range between the low risk group versus the high risk group. Then comparing the dementia signal test to the commonly used genotyping test, which is the ApoE genotyping test for dementia prediction, which is shown in red here. You can see that with the dementia signal test, with or without age and sex information, it outperformed the ApoE genotyping test. Then this model is also used for 20 years prediction or 5 years prediction of dementia risk in these data sets. And again, this is a comparison with the ApoE genotyping test on the 5-year prediction as well. And the dementia signal test outperform the ApoE genotyping test. Then we also replicate the model in a separate cohort, right? Not just on a Caucasian cohort, which is the ARIC cohort, as well as the BLSA cohort. This model were replicated in the Japanese population, working together with our collaborator, Focus Life. We also see a better performance in terms of this dementia soma signal test compared to the ApoE genotyping test. Then looking at the risk prediction versus the commonly used Alzheimer's disease markers, such as looking at brain amyloid beta plaque, as well as the common use plasma markers such as GFAP, p-tau, as well as NFL. You can see that in terms of the here, individuals that show to have a higher dementia risk were more likely to have more brain amyloid beta plaque development, and also in terms of a different risk category correlations with the commonly used plasma biomarkers for Alzheimer's disease, such as GFAP, NFL, and p-tau. We do see a good correlations between the dementia risk scores as well as this commonly used marker for Alzheimer's disease prediction. So the take-home message for this particular publication demonstrate that it is possible to develop a cost-effective, that is with minimal invasion, protein-based prognostic test to predict the possibility of dementia risk for individuals for the next 5 to 20 years. So this is our first dementia soma signal test that we have published. It doesn't require any brain imaging or invasive procedure. It's basically just a simple blood test, right? And then the test outperform this genetic risk factor, which is the ApoE genotyping test. And this test has also been validated using a different population. The Japanese population is being currently used in clinical practice in Japan. So for the rest of the world, this is still a research use only test, but this is a demonstration that it is possible to use a proteomic signatures for risk prediction. So the other example that I wanted to point out is about the questions that that you have just asked, right, regarding the storage as well as the effect of free store cycles as well as hemolysis on the SomaScan assay. So there is a recent publication, right, demonstrating what is the effect of in vitro hemolysis as well as repeated free store cycles, 3, 10, and 20 cycles on both the Affinity platform. So the author actually done the study on both the Olink platform, which is the Olink 1.5K assay, as well as the SomaScan 11K assay, to look at, if you have samples that have gone through multiple free stores and if you have hemolyzed samples, how does it affect the detection rate? So shown here are the data, right? So in terms of the SomaScan data set on the top of the volcano plot, right, under both hemolysis condition as well as repeated free store cycles, about 4% of the targets are being affected, right? And then with the Olink 1.5K assay, in terms of repeated free store cycles, about 6% of their targets are being affected. But then with the hemolyzed samples, almost near 50%, 49% of their antibodies are being affected with hemolyzed samples. And this is skewed towards a positive overexpression level that they have seen with their data sets. So this is the first study that has done by a customer where they try to look at the effect of in vitro hemolysis as well as repeated free store cycles using the 2 different protein affinity detection platform. For the SomaScan assay, they have shown that about 4% of the targets are being affected under both the hemolysis as well as free store cycle conditions. And then for the Olink, 6% of their probes are being affected upon multiple free stores, and then nearly half of their probes are significantly affected by hemolysis condition. Okay, with that, that's the end of my presentation.